Well, you will have heard a lot about the work that AWC does with animals, from Guldian finches to rock wallabies. And you may have heard us say that AWC invests more effort in biodiversity surveys than any other non-government organisation in Australia. But of course, that's only part of the picture. And we also have a national program to monitor vegetation and plants across all of our sanctuaries. And to talk about that program, I'm joined today by one of the botanists and ecologists leading that work, Jeanette Kemp. Jeanette, thanks so much for joining AWC in conversation.
Hi, Joey, and hi, everyone. Thanks for me to join.
We've got a bunch of people on the line, so it's a good turnout for episode 1 of this season. Jeanette, where are you speaking to us from, and what's the work you've been doing up there?
Yeah, so I'm at Mount Zero Taravale Wildlife Sanctuary, and we're here, myself and Regal are here to set up monitoring plots associated with the predator-proof fence where the northern bettongs are going to be reintroduced. So we've been here for a few weeks doing that. Yeah, so it's an amazing spot.
Yeah, so this is that project that we spoke about last time with Felicity. So a completely new fence project for AWC to secure a population of the northern bettong. Do you want to just describe the landscape at Mount Zero? What's the kind of vegetation like that you're working with up there?
Yes, I'm currently working in wet sclerophyll forest mainly, and a little bit of the dry country to the west. And so it's really tall eucalypt forest, crimbeas and eucalypts. That's a picture of tall grasses. And yeah, pretty dense country. And then as you go a little bit further west, so that the proposed fence area includes this sort of tall country, but also this much lower, more stunted country with grass trees, and becomes more rugged gorges. Yeah, beautiful landscape. Quite thick to work in sometimes. Running out of tape measures is a bit of a challenge.
Yeah, right. So we might come back to the inside and outside monitoring for our fence projects in a little bit. But I'm interested to hear a bit about your background, Jeanette. So you're in North Queensland at the moment, but originally you're from down south. Were you always interested in plants and wildlife?
Yeah, I guess, I mean, my dad was, and still is, a botanist in his spare time, so he used to go out collecting plants. And but the main thing is he used to take us camping, so I think I became interested in the environment early on, just because I was out there a lot. And then, you know, when I went to uni, I was really inspired on our field trips, which, believe it or not, we went out. So I went to Ballarat Uni, which is now Federation Uni, and our lecturer, Martin Westbrook, was taking us out to Nanya, which wasn't owned by Federation Uni then, but is right next to Scotia Wildlife Sanctuary, which didn't exist back then. And that's where I learned to really love, I guess, just love being out there doing fauna and flora surveys. And yeah, that was the beginning, I guess.
And for a large chunk of your career, you worked at Queensland Herbarium. What was the sort of work you were doing there?
So I was a vegetation mapper, and so using a stereoscope to delineate different vegetation types, and then going out in the field and to help describe each of the vegetation types. And I was pretty lucky to work in both wet tropics and Central Queensland coast area. And in particular, Central Queensland coast was amazing because I had to map the vegetation on the islands, Whitsunday Islands and some of the islands further south. So we spent a fair bit of time being helicoptered around and dropped off by boat into these beautiful national park islands, and spending a few days and then racing back in time for the helicopter or the boat. Yeah, it was amazing. I met some great people, and you know, there was just, we'd be doing sites up on these hill slopes on an island, and you'd hear this kind of funny blowy noise and look down and there's a whale breaching just below you. And yeah, it was a perfect job. It was great. That's
got to be a dream job, doing veg surveys on the Whitsundays. I mean, a lot of people would kill to be doing that sort of work.
Yeah, I've been pretty lucky.
And you've been with AWC now for 6 or 7 years. What was the appeal of AWC? Because you said you wanted to work for us for a long time. What attracted you to the organisation?
Oh, I guess it was, you know, even though I loved my job, there's a fair bit of bureaucracy and some of the political stuff I wasn't so keen on. I knew that the people that work for AWC just generally love their jobs and they're really dedicated, and the properties, I knew about some of the properties they worked on and they're spectacular, and I wanted the chance to work out there on those properties. So yeah, they were the main things. Yeah.
I thought I might break it up with some of your favourite plants. You sent me a whole bunch of photos, so we'll try and share some of these. This is a really cool one. It looks like it's from the arid zone. What's this one?
Oh yeah, so that's quite a widespread chenopod species, ruby saltbush, Encolina tomentosa, and it's just a group of plants. I love chenopods. They're pretty kooky. They've got these kind of fleshy, hairy leaves and a whole variety of different fruit shapes and sizes, with some of them have weird protuberances. But this one in particular I love because it's a good indicator species in our drier, sort of mallee, semi-arid areas as to condition of vegetation. So yeah, and you can eat the berries.
Oh, that's a bonus. Does that happen often when you're doing surveys?
No, not all that often. Actually, generally I try not to eat things.
For those watching at home, we don't recommend picking this going bush unless you know what you're doing. So, Jeanette, you joined AWC as a wildlife ecologist. So that was doing survey work, not just for plants, but for animals across our northeast sanctuaries in Queensland. Were you happy doing that work with animals, or were you kind of always looking at the plants that they were eating on the side?
Yeah, I loved the animal work. But that's right, I sort of was always looking around seeing all the things that could be done as far as monitoring of plants and vegetation types. And so, yeah, I guess I started to branch out a bit and always pushing to do projects looking at weeds and just vegetation. So I started setting up some projects on Brooklyn Wildlife Sanctuary, looking at differences of vegetation either side of the fence, and redoing some work that I'd done as when I was working with CSIRO about 10 years ago. So yeah, set up the projects and moved on from there. And then of course we were shifted onto a national vegetation program, which sort of started with our predator-proof fence work.
Yeah, so the kind of more dedicated vegetation monitoring has been underway since about 2016. Do you want to just list what the main priorities are? So what is the information that we're trying to collect about vegetation as part of that program?
Yeah, so one of the major things that we're doing is looking at the predator-proof fence projects across all the sanctuaries. And so the aim with that is to ideally get some monitoring plots in the early stages of the project development, either side of the proposed fence or the fence, and then use the monitoring to see how the vegetation structure and species composition diverges over time because of the differences in mammals inside and outside the fence. So that was a major one, and that's still ongoing, obviously. And then we've also started to move into developing national monitoring methods for looking at actual just broad vegetation structure and composition on all the sanctuaries, and how that might change long term with changes in our management, like taking off cattle or sheep, changes in fire management, potentially climate change as well. So that's installing a number of long-term sites on each sanctuary and monitoring target species. And then also we're developing some standard methods for monitoring weeds, even though we have been, monitoring weeds on some of the sanctuaries for a while. We're just making that a bit more standardised. So weeds and threatened species and threatened communities. So yeah, working on some standard protocols for that. And then finally, just some methods for looking at structural data for vegetation at all the fauna sites. So when you're surveying the fauna, you've got some correlate data there to do some analysis with.
And I'd be interested to hear a bit more about what you're measuring and how you measure it. And I'll share some of the photos here, but you're looking at not just the different species, but also the structure of how that vegetation is layered and how dense the different, the understory is and the canopy is and that kind of thing. Do you want to just talk us through what that work is? So what you're actually measuring in each of the plots and each of the transects?
Yeah, so it differs according to what you're doing, but so for the predator-proof fence work, we've got quite detailed plots, very involved. And so we've broken it up into species data collection, which is, well, actually, since we've got a picture there of someone measuring a tree, we've got some of the structural data includes collecting comprehensive information on tree diameters and densities. So those guys are measuring tree diameters. So that gives us an idea of, density, and then obviously it'll get resurveyed every few years and we can look at those changes. Then we collect point data along transects, and for the predator-proof fence transects, we've got 500-metre transects, and at every 1 metre point, so we've got 500 points, we're collecting height and sort of like whether they're a shrub, bare ground, live vegetation, and that sort of thing at each point. And so that gives you quite a lot of structural data. And then for the species, we're using quadrats, which is a method. So it's just so that's Liz looking at a quadrat there with not very much in it. No doubt she's found a few small things there that we can't see. She's got a good eye. So within those quadrats, we're doing, we've moved away from percent cover estimates, which lots of people use because it's very subjective, and even the one person can come up with completely different estimates if they do it twice. So we're doing presence within a quadrat. So if a species occurs in a quadrat, you just say it's there. But the downside of that is you have to do a lot of quadrat to get enough data. So we collect 120 quadrats from one plot at each of our sites. So yeah, they're the main methods.
To give people a sense of the scale of this kind of vegetation monitoring program. So, well, actually, I might just share this transect diagram. So this is, I think this is one site with, well, you can describe it, but when you calculate this, it's actually thousands and thousands of plots across the country. So these are the transects that you do, is that right?
Yeah, so they're just at the predator-proof, fenced sanctuaries. They're the more detailed plots that we're doing. So we're including every ground layer species, but each of those horizontal lines are a transect, and they go to 100 metres. So there's 5 of them, and at every metre you collect a point. You do a point and collect some structural data, and then at every 4 metres you put down a quadrat and collect the species presence data.
It's a huge task. When you think about the area, across AWC, we manage 6.5 million hectares, which is roughly almost the size of Tasmania. And you're actually collecting data on every plant species that you come across in this very detailed way. Is an inventory, like a big list of all the species, part of the work?
Yeah, So I work with Regal Jensen. He's the other, one of the other botanists working with us, and that's one of his main tasks, is to compile species lists for every sanctuary. It's still
great photo of Regal from before, in case.
That's our Regal's the Melly man in the. Yeah, I love the hat. So he, the species inventories are actually still in their infancy, because we do have so many sanctuaries, and some of them are really remote. So he started off by just collating all the herbarium data available, so all the plant collections available from each of the sanctuaries, and then obviously talking to the people on sanctuary, and then of course our own visits. To every time we visit a sanctuary, we add to the species list. So we've got some lists are more comprehensive than others. You know, the sanctuaries we haven't been to and they're pretty remote, still got a long way to go. But somewhere like Brooklyn, we've probably got, I don't know, maybe 4/5 of the species. So yeah, that's it. And he's developing a database to go with that. So yeah.
Yeah. And you shared some photos of some of the really tiny plants that you're trying to detect. So we should have set up a scale bar for this one, but
yeah,
this looks like some kind of alien invasion or something on the surface of another planet. What are we looking at here?
It's a tiny little daisy called Actinobile uliginosum, and it's probably only about 3 millimeters across and 3 millimeters high. And you can see there's actually a flower on it. I mean, in some places they're more miniaturized than others. It depends on the season. But you can see it's sort of sitting amongst some ground lichen there, and that's, you know, brown dirt there. And there's even another species behind it. I don't know if you can see further up. There's different daisy, different species, and that one's even smaller. That's full grown plant. So yeah, we find some pretty kooky plants.
Wow. You've also got some interesting bits of equipment to do this sort of work. I don't think I'd heard of a densitometer before. It sounds like some Doctor Who tool. Do you have that with you? Can you show us how that works?
Yeah, I do.
So this is just while Jeanette's grabbing that. It's a way of looking at the structure of vegetation and describing what is in a particular point. So it uses lasers and mirrors. It's almost like a periscope crossed with a laser pointer.
Yeah, so it's I'll just point this up a bit. All it is a paint stick, actually, with a laser mounted on it. And the laser is just so you get to a point and you point downwards, and it's just like it has a very fine point, so you can say that you can quickly see exactly what it's hitting. And so you write that down and you can see what the height of that bit of vegetation is. We've got some height markers on the side of the stick. So it's better than just like, you know, using a stick without a laser or using your foot, because it's just less subjective. You're less likely to go, oh, hang on, I want to get that bit of vegetation, because everyone are inherently, you know, they want to get a bit of green vegetation. So that's for. And then the actual densitometer part, which is mounted on the top, and you can buy this bit at, you know, forestry tools or whatever, and it's got two levels, a horizontal and vertical level, and that makes sure that you're holding it level, and it's got a mirror in it, so you can look in there and see the canopy without having to look up. So you get it level, and then you can say whether there's canopy or not. That's pretty much all it is. Yeah. That's
cool. Never heard of one until we were talking about it just the other day. I've got some more of your favourite plants with some images that you shared. And these are from various sanctuaries, because of course you're working all around the country. This one's a little Lobelia.
Yeah, that's just amazing. Lobelia winifred. So that's both those flowers are from the same species, and they just come in these two different colour forms. Yeah, incredible plant.
And another one here, Olearia.
Oh, they're sort of nice surprises working in the Mallee country at Yucumurra. We were finding those occasionally, even though everything else was dry. They've got huge flowers, Olearia magniflora, very beautiful. Yeah.
Great. Now you mentioned at Brooklyn, you'd actually looked at some vegetation monitoring sites there with a decade interval. So it must have been, you know, prior to your time with AWC, and then revisiting those sites. Were you able to see a difference in vegetation from the management that had been undertaken in that intervening decade?
Yeah, we were, and it kind of inspired us to set up some long-term monitoring on some of the other sanctuaries, I guess. So some of the changes were that the sort of grasses that are more palatable to cattle, like kangaroo grasses, some of the perennial sorghums, and spear grasses, became more common once we took the cattle off, which was, you know, what we expected, and it was great that it came up in the data. And also when you have, in that sort of country, when you've got a lot of cattle on country, you end up with a lot of white spear grass because they've got spiky inflorescences and not very tasty leaves. So they end up dominating the landscape because the cattle are avoiding them. So those grasses actually decreased after we took the cattle off. So yeah, that was really amazing stuff. And then of course there was some results we didn't quite expect. One of the weed species actually increased, which was just quite, it's useful for us to learn about this. Some of the weeds are probably decreasing because once you take the cattle off, you know, there's less disturbance and less capacity for weeds to invade. So we think the greater grass is getting less, which is great. But yeah, we did find that one of the other weeds was increasing, so we've got to think about how to deal with that.
So weed mapping is also part of the work, and I guess, you know, it's a really good example of how AWC's science program is geared towards how we manage the land. So, you know, something like this photo regal with, I think it's a prickly pear, is it, Jeanette? That's something that our land managers will then know about and be able to come up with a strategy for dealing with weeds.
Yeah, so yeah, that is the prickly pear. Regal doesn't look too impressed. So yeah, so we're sort of coming up with some standardised methods for measuring weeds, and so it sort of starts off with we've got these eco health plans, and related to that is our weed strategies for each sanctuary, which are just being developed. And as part of that, we'll have a number of target weeds, and then we'll, you know, start rolling out ways of monitoring those targets to make sure that we're actually making an impact with our control measures.
Great. Jeanette, I'm also interested with the inside and outside fenced area projects, whether there are clear differences that you can detect there already. You mentioned that some of the plants are probably favoured food species for the mammals that we're reintroducing, things like, you know, bilbies and some of the betons maybe. Are you able to detect that in the data?
Yeah, so we've there's a heap of sanctuaries we're just establishing sites that will be remeasured in, you know, 5 or 10 years' time. And they were because they were, you know, newly fenced areas or fence proposals. With Scotia and Yucamara, they'd already had animals either side of the fence for 10 years. We were able to immediately compare the vegetation either side of the fence, even though you can't be 100% sure that those differences weren't there originally with, you know, that particular example. But yeah, so we have noticed some differences. So in particular, plants with bulbs and fleshy leaved plants such as the little chennapods. So you know, one of the chennapods, one of the many kooky chennapods that are pretty cute. So some of those species become less abundant within the fenced areas. They're still present, but they're less abundant, especially larger forms of them. So there might be plenty of little seedlings, but there's less of the larger ones. And yeah, that's a lovely Mariana coronata, great plants. So I guess where, you know, you look at that and the reintroduced fauna, they're obviously creating a different environment within the fenced area. They're digging holes, digging up bulbs, eating seeds, changing kind of moisture and you know, landscape properties. So there could be longer, although we haven't picked it up yet, there could be longer term changes in density of shrubs, for instance. And yeah, so that's what we're hoping to pick up
Yeah, and I think it's again just an illustration of the AWC's dedication to long-term data sets. So you're establishing these plots now at some of the fences that they're just building in New South Wales and at other sites, but they're sites that will be continually monitored at intervals where we can pick up long-term changes in how that vegetation adjusts to the reintroduced animals. Really important for understanding how it all fits together.
Yeah, I mean, there's so much still to learn about it, and coupled with that will hopefully be some more diet studies on the different animals. But of course, it's quite complex. It's not just about what the animals are eating or disturbing. It's also there's differences as far as where we generally remove some of the larger macropods within the enclosures, and then of course all the feral herbivores get removed as well, and then outside the fence there are still some feral herbivores. So it's differences between those mammal assemblages really we're looking at.
We've got a question that's kind of relevant to this, so I might just ask it now. But with the long-term vegetation plots, is there a typical interval that where we plan to go and resample those sites, or has that not yet been established?
It's not really in concrete. We haven't done any resurveying. It's all about resources, really. We'd probably like to resurvey them every 5 years, but it may end up being a little bit longer than that, just due to resources. But you need that amount of time just to pick up the changes in the data.
Yeah, and it's a good point. We've got it's basically you and Regal as dedicated botanists on the AWC team, plus a bunch of volunteers who always help a lot with these things. But 30 different sites to get around to now across 6.5 million hectares. It's a really big job.
Yeah, and that's why so we're also sort of developing some monitoring to look at changes on all the sanctuaries in vegetation. When you change management, there's going to be changes, and so because we don't have 30 botanists, we've narrowed it down, or hoping to narrow it down to a number of indicator species, particularly in the ground layer, and they might be dominant species that the managers have noticed change with a particular management action, or some of the weeds, and so you just have this smaller set of target species that any of the wildlife ecologists can collect that data.
When we were preparing for this, I asked if you had, ever come across something that was unknown to science or a completely new species of plant. Not yet, Jeanette?
Oh, not on the sanctuaries. But we, there are some pretty rare plants that we come across. So Regal in particular spends a bit of time, although often on his weekends, searching for some of these things. And it was actually a mate of his that found this one on Brooklyn. This is, it's actually in the Ericaceae family, so the heath family. So it's Stiphelia geniculata. And it's only known from Brooklyn. There's only 2 or 3 little spots on Brooklyn that it occurs in this really kind of restricted habitat. So that, only place in the world it's known is Brooklyn. And so, in fact, we haven't even got brilliant photos of it yet. So we need to go up and do that. But the habitat is these steep ridge lines with boulders, granite boulders. I think you can see that that's a shrub in amongst the boulders there. So that sort of stuff is really important, like just knowing where these plants are on sanctuary, so we can keep an eye on them, make sure we're not, damaging them with fire or things like that. So yeah, we do come across, and also Regal spends a bit of time looking for orchids, which, believe it or not, even though they're a popular plant, they're very rarely collected up in this area, probably because, they're only out for a short period of time. And so we're still finding quite remarkable range extensions in, areas that are only 60 kilometres from Townsville. So,
wow. So much more to do. And then finally, Finally, Jeanette, I was also interested in your favorite plant, or whether you had one you were especially fond of or that you like coming across. And you mentioned a grass, and I think grasses are probably not celebrated as much as they need to be. But the real hardcore ecologists always talk about grass as such an important part of telling the health of an ecosystem. Can you tell us what this one is?
Yeah, so it's a kangaroo grass. So I love grasses, and everyone should love them. So this one in particular, it's one of the most widespread grasses in Australia, and I've got a heap of it growing in my garden. It's a pretty strange garden, and I like it because it's often a good indicator of health, of ecological health. So, for instance, at Brooklyn, once we've taken the cows off, I think we're getting a bit more kangaroo grass. It's quite sensitive to disturbance, and in some places it's been almost completely wiped out. Like around Melbourne, it was once very common, and now it's just restricted to these tiny little railway reserves and cemeteries and things like that. So it's almost become a rare plant down there. Yeah, I just think wherever I see it, I feel calm and relaxed and happy, and know that it's somewhere that's in good health.
Good omen. Jeanette, thanks for describing the work that you do. We might come to some of the questions now. And we've had people asking about actually that last picture of the specimen of grass, and whether you actually collect herbarium specimens as part of the work to catalogue species. And I might add to that, has technology changed the way that you do that?
It hasn't really changed much at all with collecting specimens. So the herbaria around Australia, there's one sort of major herbaria in each state, and they've each got a giant collection of plants, including plants from way back when Cook was sailing around Australia, and yeah, pretty much collect them exactly the same way that they did then. And so we do collect as part of our work, mainly if we unsure of the identification of a species, or if it's just a really great example of that species from that area, with good flowers and fruits. But the other thing it's good for is if you send in specimens, you've basically got a permanent record with a dot on the map for that plant all over your sanctuary, all those different plants all over your sanctuary. And if they have name changes or anything like that, we've got that data, and so we can keep up with that. So we actually try and get a specimen of each species from each sanctuary sent to the herbaria. We've still got a long way to go, and Regal Jensen, who was with me, that he spends a fair bit of time on that, and that's pretty important. So this is you can see that's a plant press.
Right.
Probably almost the same as what they were using in the old days. And so it's basically just, pressing several pieces of a plant in sort of 2 or 3 sheets and drying them as quickly as you can. So you either put it out in the sun, or you can put it in a plant dryer, which is basically just a heater in a box. And then it gets sent to the herbarium. And so we also collect DNA material, which is just a few leaves put in a packet of silica so it dries out quickly, and that goes with a specimen. And it actually helps when we collect DNA. It's more likely that the herbarium will then keep the specimen because it's coupled with a DNA thing, and it's that's advantageous for us because we want a record of that species from the sanctuary. But then also, of course, it's used for taxonomic purposes. So they actually do want that DNA. And then, of course, Regal takes photos of all the plants. So he's getting pretty good at taking some pretty specky photos of all the collections. Yeah. And so they're now, most of the herbaria are now actually keeping those photos as well. And, I guess they hope to eventually link it all up so you can view the photos and the specimen online. And then, of course, we can use those photos on our, you know, when we're out in the field, because we used to carry around great boxes of reference sets of specimens, but we've found now it's just easier to have the photos on your iPad or your iPhone. You can just sort of flick through them.
There's, I just found one that I missed, so I might share that because that's such a beautiful photo. But yeah, as you say, you know, that kind of technology, having that information in the field is really streamlining a lot of the work that we do, not just with plants, but with wildlife surveys as well. We've talked a lot about camera traps before on here. So, you know, it's interesting, I think, in botany, some of the technology, plant presses and stuff, as you say, that's what Joseph Banks would have been using. And, you know, for 100 years, it hasn't really changed. But then with photography, imaging, you know, DNA, there are aspects of it that are, you know, really cutting edge technology.
Yeah, and that Dodonea, it's a little hot bush, which is just such a beautiful plant, and that's growing on Mount Zero Taraval, and it's actually quite a really weird isolated patch of and it, you know, it grows a lot further south as well. So it's a possibility one day it might end up being you know, called a different species, and so having that collection in the Queensland Herbarium means we can keep track of any name changes in the future.
Yeah, great. Okay, we've had another question here just asking about, well, bryophytes, I guess. Do you include lichens and mosses in your vegetation surveys? How small do you go?
I think I might have an idea who that question's from.
Okay.
Yes, unfortunately we don't do bryophytes because we've just got so much to do, but we hope to do that in the future. And, you know, obviously they're a huge part of the flora.
A couple of other questions about how much difference fire can make. We've talked about fire management at different sanctuaries in previous webinars. So a lot of AWC's land management work, particularly in Northern Australia, but also at our southern sanctuaries, is re-establishing healthy fire regimes. And typically that's about, you know, low intensity burns, which, you know, can be more frequent or less frequent, but, you know, often it's more regular, low intensity fires. How much does that affect the structure and the species composition of plants?
Yeah, well, I think it varies according to the area, and that's one of the reasons we're starting to roll out some of this broader monitoring, because we actually don't have a really great idea yet of those sorts of changes. We do know in some areas that over time, you know, obviously if it's burning too hot, then you'll end up with less trees because the trees keep getting knocked back. Yeah, there's lots of differences that we don't know yet. Obviously, in the short term, it affects the fauna. So that's why we actually have these, or starting to have these plots set up at all the fauna sites. Every time they do these surveys, they can get a record of the, you know, height and density of the ground cover, and they can use that to correlate with their fauna abundance and presence. But yeah, a lot of unknowns actually.
Yeah, and I think, you know, the long-term aspect of it is important to note here, that, you know, we're really establishing this framework for monitoring plants in a way that will tell us things in a couple of decades. So, you know, it's a long process to find out some of this information. Another question, we've actually had a couple of questions about imaging and mapping. So someone online is a geographer. Thank you for listening. And just asking about how you use satellite imagery. So I think you mentioned earlier the, what's the tool called that you look through to look at satellite images?
That's a stereoscope. So, well, no, that's just looking at aerial photos. So that's, we as far as vegetation mapping goes, we're still using the main method, which, well, Queensland Herbarium uses it, which is actual hard copy aerial photos, which come in sets with overlap. And so you can use this stereoscope, which enables you to see the landscape in 3 dimensions. and it makes it much easier to map out the different vegetation types. So we actually have one of our staff, Peter Stanton, that's his main task, is vegetation mapping. So that's the way he does it. And then it's, it's drawn onto the aerial photos, and then it can be digitized. And we've got some semi-automated methods for digitization as well. So obviously that can be combined, and we're actually trying to move towards a more combination of methods where we use that plus manipulation of satellite imagery, and end up with a product that's a little bit faster to make, but still the same quality. So yeah, we're starting to talk about that now. Yep.
Yeah, and interesting. You mentioned Peter Stanton, who is actually one of AWC's longest-serving staff members and an absolute expert at that method of stereoscope vegetation mapping. So yeah, he's contributed a huge amount to our understanding of vegetation across various sanctuaries. So we thank Peter for all of his time with AWC. Okay, two more here. So one is someone who's studying ecology or studying science at the moment and interested, I think the question is saying that, botany sometimes gets sidelined. People are more excited about animals. What's your pitch to someone who's at that stage in their career? Why should people be interested in botany?
Oh, absolutely get stuck into botany, because I reckon even though your lecturers might say there's no opportunities, I reckon there's loads of opportunities, because there's actually not many people out there that really know their plants, and it's always sought after. So if you can make yourself a niche in that area, you'll become sought after. And plants are just fantastic. They're much more interesting than animals. You can just understand a lot about the landscape if you know a little bit about the plant. So to me, they're hugely important.
Yeah, great. And then a few people interested in volunteering. So, if people are particularly passionate about this work and would like to help out, is the best way just through the volunteer portal on our website? There are opportunities for volunteering on this kind of work, Jeanette?
Yeah. Oh, definitely. Absolutely. So particularly if you're interested in plants, that would be really great. We use volunteers a lot, and we really enjoy having them. It makes the work so much easier. So you can either, email me directly, go through the AWC. Either way is fine. So yeah, definitely with us.
Yep, and we do have a dedicated portal. So for any volunteer opportunities, you can sign up at our website. So that's australianwildlife.org, and you can scroll down to find the volunteer portal. If you sign up there, then you'll be emailed out every volunteer opportunity around the country. So it's a great way to get some hands-on experience. People also asking about whether citizen scientists can help with this kind of work. So a bit of a related question, I guess, but there are more and more opportunities for people to get involved in that kind of work, I think. Yeah.
And, we couldn't do the work we do without volunteers helping us, basically. We probably do 3 times as much work when we have volunteers.
Jeanette, thank you so much for your time today. And I hope the rest of your time at Mount Zero goes well. Maybe you'll find something new up there.
Thanks, Joey. Yeah, there's always that possibility, and it's just lovely to be out here working in the bush.
Half your luck. And thank you to everyone who's joined to watch this webinar today. It's been great to get back on the airwaves and see you all again. Next week, we have an episode coming to you from Scotia Wildlife Sanctuary in Western New South Wales. So we'll have our ecologist out there, Tarly Moyle, and the sanctuary manager, Hamish. So they'll be talking to us about their year at Scotia. They've settled in at a time when Scotia's come out of a really severe drought, but they've had quite a bit of rain over the last few months, and there's been wildflowers and the first signs of recovery in that landscape, which is really exciting. So we'll talk all about that, as well as the mammals that have been reintroduced there and how they're going. So thanks everyone for joining me. As usual, if you've been inspired by the work that AWC does and hearing about this really ambitious vegetation monitoring program that Jeanette is part of, please consider making a donation. You can do that at our website, australianwildlife.org, and just click on the donate button. Also, make sure you signed up to our email newsletters. So you can also subscribe at our website, australianwildlife.org. Until next week, see you later. Thanks again.